In this chapter, chapter 12, we're going to be looking at the application of all of this information we've learned about chromosomes and cells and look at DNA technology. This is a great chapter to really look at how we can use this information and why is it important to us. So we can start with the idea of DNA profiling. This is probably something you've seen on TV shows or movies, how they gather a DNA sample, maybe from a crime scene, and then they analyze it and try to determine what individual it came from. So DNA profiling is used a lot to indicate the guilt or innocence of somebody under investigation. So shows like CSI or NCSI utilize a lot of this DNA profiling. But other than that, DNA technology is used for other things, such as the creation of genetically modified crops or things like GMOs, which we're going to talk about later in the lecture. Also can be used for great things such as the identification and treatment of genetic diseases, like how we talked about Huntington's disease and how a person can be tested to see if they genetically carry the dominant or the recessive trait. These are all examples of DNA technology. So biotechnology is a broader concept and really looks at the manipulation of living things. So we've been using biotechnology for thousands of years. When we grow a certain kind of crop in a certain way, that's biotechnology. When Gregor Mendel was manipulating the pea plants, that is biotechnology. When you pick this particular animal, this sheep and this ram, and you mate them together because you're looking for a specific trait in the offspring, that's biotechnology. Today, we've kind of advanced biotechnology because we're able to look at it from the genetic level. So we can manipulate living things at the genetic level. So instead of just saying we're going to take this sheep and this ram, we could harvest the egg from that sheep, we could harvest the sperm from that ram, we could alter them and then put them together in a way to create a clone or to create a super sheep that would have the specific traits that we're looking for. DNA technology also allows us to move specific genes from one organism to another. So we can literally cut and paste genetic information, creating a variety of different kinds of organisms or organisms that have a specific trait that you might be looking for. If we were to do this, if we were to combine genetic information from more than one organism, we call that recombinant DNA. So if we took a slice of DNA from a human, for example, and we cut that and we put that into the DNA sequence of a bacteria, then that bacteria would then contain recombinant DNA. So it would be two different sources of DNA within that same molecule. The process of doing that is known as genetic engineering. And this is us meaning humans, directly utilizing, manipulating, changing genetic material for our own personal gain. And it's amazing, the field of genetic engineering and what we've been able to do, what we will continue to do in the future in terms of altering genetic information. Like these fish. You can read more about these fish in your book, but the glowing trait of these fish is not normal. This is genetic manipulation. This is cut and transferred DNA, recombinant DNA, from one organism that produces bioluminescence naturally into another organism that doesn't. So in this case, this is really more of an aesthetic thing of making fish that glow in the dark. Maybe we could make humans that glow in the dark, or we could make other living things that glow in the dark. Not necessarily a practical thing, but the point is that it can be used. There are many more examples of the practical use of cutting and pasting DNA from one organism to the next. This has led to what we call farm animals, PHARM animals. And these are living organisms, usually animals, but not always, living organisms that have been manipulated at the genetic level to produce something for us. Just like we've taken, the wild cow or the bovine, and then we have domesticated it and made it useful to us through selective breeding processes. So this is kind of the same thing. This is taking a living organism, genetically altering it so that it produces something that we want. An example of that is our source of what's known as humulin. So prior to 1982, a person who had type 1 diabetes, their only option really for insulin was bovine or horse insulin. So insulin that came from a dead horse or from a dead cow. So somebody who has type 1 diabetes does not produce enough insulin naturally from their pancreas. And so the treatment for that disease was to harvest insulin from other animals that were usually slaughtered for meat or for other purposes. They would extract out the insulin from the equine insulin from horses and the bovine insulin from cows, and then they would inject that insulin into a human. The problem is twofold. One, it's extremely expensive, and two, it's not human insulin. It's not the same thing. So it was a treatment, but it really wasn't a very good or very successful treatment at the time. So here's what genetic engineers did. They were able to isolate the gene that's on, that humans have, that codes for the material to make human insulin. So remember that Watson and Crick, how they came up with this image of what the DNA molecule looks like, and I told you about how that completely changed the medical field and the technological field in terms of genetic information. So here's a perfect example. You take a human gene, like the gene for insulin, and you cut it out. And we're going to talk about how you cut DNA, but you cut that DNA out. Then you have another organism, like a bacteria, for example, and you cut that bacteria's DNA and insert the human gene for insulin into the bacteria's DNA. And remember when we talked about the mRNA codes and amino acids and how they're all the same, and the letters of DNA for all organisms, it's all the same? It works. This absolutely works. So you have now a bacterial strand of DNA that has a human gene in it. And the DNA in the bacteria don't recognize it as anything different. So then when the RNA polymerase comes along and it wants to make a piece of RNA, it will transcribe the human gene, making a piece of RNA that will then leave the nucleus and go out into the cell and be translated into human insulin. It's literally the same thing. You can't tell the difference that if it's coming from a bacteria or a human. This absolutely has changed the world, especially for people who have type 1 diabetes. So today, these people are able to get human insulin, not equine, not bovine insulin, but human insulin. It's still a little bit expensive, but definitely more affordable for people to have. So here's what that humulin looks like. And if you look a little closely here at the label, you will see that it says that this is recombinant DNA, that this is how this was made. It was literally cutting and pasting pieces of DNA together to get what we wanted. So today, this humulin is continually produced in these gigantic fermentation vats. It almost looks like where they make beer in a microbrewery. And they're able to extract this material, and then is filtered out to harvest the human insulin. So here's what it looks like. These pharmaceutical companies are able to literally make human insulin from a bacteria. So DNA technology, it can be used for many medical purposes to make supplemental material that some people might be missing. For example, human growth hormone, HGH, which is given to children who don't grow at a rate fast enough or at an adequate rate, so they can inject themselves with HGH. It can be used for the hormone EPO, which stimulates more red blood cell production. What's interesting about those two is both HGH and EPO are used artificially as a form of steroid in sports enhancement or physical activity enhancement. We'll talk more about that later. But DNA technology can also be used in developing vaccines as a way to protect us. We can alter a virus to contain the genetic information that we need that can then lead to the person who's injected with that virus being immune to an infection. This book, The Immortal Life of Henrietta Lacks, is an excellent book. You don't have to be a science person to understand this, but it's about the history of a black woman who had cervical cancer and went to Johns Hopkins University for a procedure. And in that procedure, they removed the tumor that was on her cervix, replaced it with a type of cancer treatment at the time, which was basically putting radioactive material directly onto her cervix. And then eventually, unfortunately, Henrietta Lacks passed away from complications due to her cervical cancer. But what's interesting is that the tumor that they removed was studied in a lab. Cancer cells continually go through mitosis. They don't know to stop, so they just keep making copies of themselves. Well, Henrietta's cells were very unique, and they continued to grow inside of a culture dish in a laboratory. So they started to experiment with Henrietta Lacks's cells, and they started using them for research at Johns Hopkins University. And other labs were like, hey, what is the cell strain that you have that continues to reproduce? Can we have some of that? And they began to sell those. And they themselves, Johns Hopkins, received many awards, including Nobel Prizes, and other facilities paid money, all for these cells that were taken from Henrietta Lacks without her permission. And her remaining family members, her 3 children, did not receive any financial compensation. That's what this book is about. So it's a story about medical ethics. It's a story about a poor black woman and how she was at the time taken advantage of, but there were no rules in place really to protect her. And it's also a story of how the author is trying to get financial compensation for the children of Henrietta Lacks. So that brings us to the controversy of genetically modified organisms. So GMOs are any type of organism, it could be a plant and it could be an animal, whose DNA has been modified or changed. And it's really an interesting time right now because GMOs are not necessarily new, but it is coming to light how much GMOs are used in our society. We've all eaten GMOs. We've all had genetically modified food in some way, but we just didn't know it. So now the pressure is being put on a lot of these companies to tell us that this is a genetically modified organism or that this animal ate genetically modified food so that we can make informed decisions. So this really brings a political realm to the world of biotechnology as well. In the United States, roughly 1/2 of corn crop and over 3/4 of soybean crops are all genetically modified. And since we happen to live in a state that is predominantly run by agriculture, growing corn and soybeans, this really has a major effect on us. And it isn't all bad. I think a lot of times people think that all GMO food is bad, which there's still an argument for that. You know, do we really understand? Is it a good thing? Is it a bad thing? But it's something to think about in terms of, for example, the European corn borer. So the European corn borer is a moth, and the moth would come around to the corn plant and would lay its eggs on the ear of the corn, and the larvae would get underneath the husk and eat away at the corn and basically ruining the corn cob. So here's a picture we see of that. So you can see the ears here, and you can see here is a larvae that is eating away. Now the farmer cannot use this corn now because it's been contaminated, it's molding and it's rotting and being eaten away by this larvae. So what would a farmer do? Well, in the past, they would spray it with chemicals. So they could spray this entire crop with chemicals and it would kill either the moth or the larvae after that larvae had been raised. So it kills the pest, but it's adding chemicals to the environment, which other animals or other plants might absorb and cause a lot of damage. So with genetic modification, instead what we could do is we could change the corn at the genetic level so that the European corn borer can't eat it. Or actually what happens is that when the larvae eats it, it fills up the stomach and causes the larvae to die. So it doesn't contaminate the corn. Now we don't have to spray it with the pesticides. So the question is, do we leave corn natural but spray it for pesticides, or do we genetically alter the corn so that we don't have to spray it? That's the big question. Another example of food that has been altered is golden rice. So sometimes we alter the food to make it more nutritious. So in some places in the world, rice is the predominant form of calorie or starch intake, but rice itself does not have very many vitamins to it. So what scientists have done is they have genetically modified the rice to contain more vitamin A. So they can make it a super rice, or what they call golden rice. And you can see that here in the picture. So people eating the golden rice are going to be getting vitamin A in addition to the starch and the calories in the rice that they eat. So the question is, do we take this knowledge and this technology that we have to make better food, or food that is better for people, and feed those people, starving people who don't have access to better food? Do we use that to feed these people, or do we say that there's too much danger in producing this type of food? And we're going to stop with that and continue that discussion in class.